item on the agenda is to remind everybody that we have an open assignment right now. If you haven't already completed it, it closes this evening. It's 11:30 p.m. So it's part of our third test. Make sure you go out there and do the 40-point worth of our third test, which is available on Moodle now, and it will be closed at 11:30. So at that time, all open tests will automatically submit. So make sure you give yourself plenty of time to complete that if you haven't already done so. All right, so then what I'm going to do is probably Wednesday is post the remainder of our test 3, which will cover the things on polymers and plastics and maybe some of this biochemistry stuff, I don't know. Then after we finish up this week, and I guess Monday of next week will be our last sad day of lecturing. And then after that, what we will do is we will, after we have some turkey, I will have a final on Moodle for you to complete, and that will be that for that. Okay, so let's just begin with a little recap of where we've been and what we're up to. We have begun, we're still generally talking about the chemistry of carbon, but now we're getting specific with the type of carbon chemistry that we're focusing on, which is the basis for living systems. And we began with a discussion of what we call carbohydrates. Carbohydrates, as the name implies, have carbon, hydrogen, and oxygen. That's the only components or materials that are in carbohydrates. Carbohydrates, of course, are very fundamental. As if we didn't know it before, we gave some indication last week as to why carbohydrates are so fundamental to living systems. They literally become the fuel of living systems and the structural components of, in particular, the plant side of the hierarchy. The animal side uses inorganic structural forms and a skeleton, a little bit different than the plant side of the hierarchy, which uses the cellulose-based form of structure, which we will come to probably later today. So in any event, we have this carbohydrate basis of the plant life, which is, it begins in the form of what is known as glucose. And then glucose is kind of like the building block, the elemental form of plant life. It is manufactured by plants in the photosynthetic process, which we spent a great deal of time last week describing and discussing. And the way in which plants build glucose is by taking carbon dioxide and water from their surroundings, combine that with some sunlight to input energy to build the glucose, and then they put these components together to build individual monosaccharides known as glucose. Now, glucose comes in a variety of isomeric forms, and those isomeric forms often combine to make disaccharides. We talked about some disaccharides last week, including lactose, maltose, and sucrose. We talked about another monosaccharide known as fructose, which turns out to be how fruits and even a little bit in vegetables, how these have certain appealing flavors to them by the amount of fructose that is contained in them. All right, and then that's kind of where we were. We were at the point of looking at mono and disaccharides. Then I introduced a few artificial sweeteners, which are non-organic based molecules. Well, actually, sorry about that. Many of them are based in organic chemistry. They are not however, traditional carbohydrate form. And I indicated to you the reason is such because that in the non-carbohydrate form, our body doesn't store them for later usage in times when we are low on carbohydrate fuel. Our body has a mechanism to store it in the form of fat, which turns out to be bad because we certainly are not as it was hundreds of thousands of years ago when we really needed to keep storage. We really needed to store carbohydrates in times when we were very far apart or not commonly finding food sources. So now, since we have readily available food sources, we all tend to ingest a little bit too much carbohydrate. Then this carbohydrate is converted into storable forms, which turn out to be not too appealing to us. So humans have come up with artificial sweeteners that still give us the pleasurable aspects of our food sources, but do not store on our bodies in the same way that the traditional disaccharides that we use to sweeten foods commonly do. So I think what I'm going to do with that is to go to where I was presenting some of these ideas, and I think that's where we concluded, and I do believe that that's the first thing we're going to be looking at here is a picture of that thing we ended with, and what that was, excuse me, here was this slide that we're looking at here, slideshow from the current one. And this is a molecular model, a computational chemistry program which mimics the behaviors of nuclei and electrons around the nuclei, showing the connectivity between them. And this is a molecular model of not the electrons, but just the connections in kind of a tube representation of the carbons, hydrogens, and oxygens that constitute the disaccharide known as sucrose. Now, if you remember from our earlier discussions, disaccharide is two monosaccharide units fused together with an oxygen linking them together. The two monosaccharides in the structure of sucrose are glucose and fructose, bonded together by a oxygen linkage to the two monosaccharides. And I showed this cartoon before. What my computational chemistry program can do is now over this framework. So this is what the prediction is for the locations of the nuclei. The little white tips are all hydrogen atoms, and then the other connecting parts are either carbons or oxygens. The oxygens are slightly different color than the carbon connections, so you can discern where they are as well. But over this framework of the location of the carbon, hydrogen, and oxygen nuclei, I can lay a skin, if you like. I can put a derma, and the derma represents how the electron clouds will buzz around the nuclear skeleton. So again, here's my nuclear skeleton, and now I can put the cartoon of the electron cloud around them. Now the electron cloud is color-coded. And what the color coding of it means is if charge is evenly distributed in a certain region of the cloud displays a green color. So green means there's pretty much a balance of positive and negative charge from the relationship of the electrons and the protons in the structure. These are regions of the molecule that appear neutral. Now then, the bright red regions of the molecule are regions of the molecule where there's an excess buildup of electron density and where the electrons are in great likelihood of being, the red spots indicate these spots on the molecule are negatively charged. Then you'll see a few little spots elsewhere that are displaying blue. What it means in the blue regions of display is that there's a paucity of electrons. The electrons are spending very little time in those particular locations. So what's going on there then is that the nuclear positivity is kind of shining through the cloud. So the blue spots are where the nucleus is more likely to let the positive charge dominate. The red spots are where the electrons have the negative charge dominating. And then the green spots are electrically neutral, where there's a balance of the positive and negative charge. Now, here's the basic idea of why this is important to us, this model of what's going on here. Now, remember, this is nanoscopic. This individual sugar molecule is only about 10 to the minus 9 or 10 to the minus 8 meters across. Super, super tiny little framework here. But when you chew these things up and they're floating around and coming in your mouth and going towards a receptor in your mouth that says, ooh, that's sweet. What the receptor in your mouth has is, it is also, of course, a collection of atoms that are combined together. And those collections of atoms are also folded and bent and shaped around one another so that they create a cage that binds this molecule very specifically. And when this molecule binds into the particular shape of the so-called receptor molecule that is one of our taste buds, we get the sensation that, ooh, that's sweet, and I kind of enjoy it. So when that occurs, certain endorphins are released in your body, certain electrical stimuli are enacted in your body to give you the pleasurable sensation of that tastes like something I really like. Okay, now, so again, let's just turn the electron density off, and here's the nuclear framework. You turn the electron density on, and that's what this looks like to the binder molecule that tells you that this is a sweet molecule. So now on this next slide, what I'm showing, okay, I point out that the blue is the highly positive region, the red is the highly negative region, the green regions are electrically neutral, we'll say. Now what I have listed on this next slide, first I define this thing called a qualitative structure activity relationship. And all I'm saying here is if I look at other materials that may have a similar appearance to the way electrons are distributed around them, that maybe they could bind into this receptor and have a similar pleasing effect when you ingest them in your mouth. So here are a collection of actually more than 6, there's 8 other molecules that you see on here. The one in the upper left-hand corner, sucrose, again, is table sugar. Then I have the 2 monosaccharides, fructose and glucose. Then I have the disaccharide maltose, which, as if you recall from what I talked about before, that is called malt sugar. This is what cereal grains and wheat and things like that have in them to give them some slight sweetness. Now, believe me, grains are not particularly renowned for being sweet, but this is the disaccharide that they contain that would give them the degree of sweetness that they do possess. After that, I have saccharin, cyclamate, aspartame, and ethylene glycol. Now, saccharin, cyclamate, and aspartame are artificial sweeteners. They are not carbohydrates. So your body does not process them, like carbohydrates. And your body will not store them in tissues of the body in the form of fats because it doesn't have a mechanism to do so. Now, the last one, as I talked about before, ethylene glycol, that is an ingredient in antifreeze, it turns out. It was commonly used it's actually an industrial chemical used in a whole bunch of industrial processes, but most familiarly and most notoriously, it is something that has often been put in antifreeze so that it keeps your radiators from overheating if there's too much heat in the engine, and it also keeps your engine block from freezing if you drive into a particularly cold region. So here is this same picture that we saw 2 slides back. This is the way, after I put the electron skin, if you like, the distribution of electrons around the framework of this disaccharide sucrose. It has these familiar, at least now, red spots and blue spots that, again, when placed in your mouth and put into a binder or a receptor molecule, which is designated biochemically to signal a sweet sensation. Now, if we look at the other ones, we will note that they are not exactly the same. However, yet you can note that there are, in many regards, similarities to the look of fructose, similarities to the look of glucose, and certainly similarities to the look of maltose. So you can understand that the same location in your mouth that would bind a sucrose molecule could also interact with a fructose, glucose, or maltose molecule, and in many regards get the same sort of sensation that you would get from table sugar. So when a dietary company is looking for artificial sweeteners, they're going to look for patterns of behavior of the electron density around the nuclear framework that mimic these sorts of look, so that now if they put these in the food instead of putting disaccharides like sucrose or high fructose corn syrup in there, they would still taste sweet yet not have the caloric content. And we'll say more about the calories involved here a little later when we talk about other carbohydrate forms. So here is what the electron density around saccharin looks like, what it looks like around cyclamate. It does appear quite different, but turns out this is a very sweet tasting artificial sweetener. This is what it looks like around aspartame. And again, maybe what I can do is go down here and hide my little share button that I didn't know I could do. And then finally, as I talked about before, the ethylene glycol is not an artificial sweetener. It is, however, a molecule that is notorious that since it was contained in antifreeze for years, that children or more likely even pets, when antifreeze leaks out of a car and drips onto the driveway, it was reported many times that pets would lick the antifreeze that spewed out of a car radiator because it had this sweetness sensation, and you could see that it does have characteristic electron density around it that it could bind into a sweetness receptor. And it turns out that when you ingest even a small amount of ethylene glycol, it can be lethal after that ingesting of this material. And actually turns out it's a very painful death that happens from the crystals that are formed in your kidneys from the ethylene glycol that solidifies in your kidneys. Okay, so on this slide, what I show you is the rating that the dietary industry has assigned. Now, how they come up with this rating, I can't say 100%. A lot of it is probably not objective, it's subjective. So what they would do is get a panel of people, sit them down in a room, feed them these various sweeteners, and then have them rate them for sweetness. So somehow the industry has decided how to determine the sweetness of a variety of artificial sweeteners and a variety of mono and disaccharides. So what they then did was, based on the rating that the individuals who were subjects of this test, they decided, okay, how does this material compare to sucrose? Is it half as sweet as sucrose? Is it twice as sweet as sucrose? And so forth. So they just arbitrarily assigned the number to sucrose of 100 for its sweetness. Now, again, this is an arbitrary assignment that was attached to what sucrose seemed to be in terms of how sweet it tastes when you ingest it in your mouth. So then what you would do in this survey is you would take another sweetener and you would arbitrarily decide if it was sweeter than sucrose or less sweet than sucrose. If it's sweeter, you give it a rating over 100. If it's less sweet, you give it a rating under 100. If you think it tastes 3 times as sweet, how can you do this? Get the fact that it's 3 times as sweet. If you eat 1/3 as much of it, you weigh out a certain amount of sucrose and it has a certain sweetness to it. If you have 1/3 as much of another material, then that would be 3 times, and it has the same level of sweetness, then that material is 3 times as sweet as sucrose. If you have to ingest 4 times the amount of some other sweetener as you do sucrose, then it is 1/4 as sweet as sucrose. So then it would get a rating of 25. So after this was done with probably thousands of people and very carefully tabulated, the industry of the foods manufacturers and such came up with the following ratings: that glucose was about 3/4 as sweet, because that's what that number indicates. The size of that number is 3/4 as big as sucrose, so it's 3/4 as sweet as sucrose, if you like. So you'd have to, if you wanted to think about how much you'd have to put in, you'd have to put in 1 1/4 as much glucose, stir it into your iced tea to have your iced tea taste as sweet as does sweetening it with sucrose. Now, maltose is only 33%. Now, again, maltose is the grain sugar. And I think we're pretty all aware that grains are not the sweetest thing out there. So the sweetening agent in grains is not nearly as sweet as it is for our normal sweetening materials. Lactose is milk sugar, even about half. Note that number it's half as sweet as the malt sugar. Now, going the other way, what we will find, fructose is way sweeter than sucrose. So what the industry has determined is that it's cheaper to sweeten many popular sweet things that we ingest, not with the same kind of sugar that you put in your coffee in the morning. They sweeten it with what is known as high fructose corn syrup. So certainly in all candies, certainly in most soft drinks, Gatorade, all those sorts of things, this is what they put in them to sweeten them. And it's, as you can see from the number rating, they can get about twice as sweet as sucrose. So essentially what that's saying, you can use about half as much fructose as you would have to use glucose in order to get the same level of sweetness from that particular carbohydrate. Now again, where do they get this? Mostly from corn. So they distill corn down. Don't ask me, I'm not an expert, I don't know exactly how they do it, but somehow out of corn, they get high fructose corn syrup, and they add that into a wide variety of the products that we ingest, particularly things that we like that are sweet. Okay, then as some of the artificial sweeteners go, you can see cyclamate that number tells you it's 3 times as sweet as sucrose. So you only have to use 1/3 as much of it as you would have to use normal sucrose. It also has the benefit of not being a carbohydrate, so our body won't store it, and so it's not fattening as sucrose is. Then we can go to aspartame, another artificial sweetener, 15 times sweeter than sucrose. And then finally, saccharin, 35 times sweeter than sucrose. All right, so let's then look at the most important thing that plants do with the monosaccharide glucose. And again, as I mentioned, first, sunlight, carbon dioxide, and water are combined in the miracle of the life of plants, put together to make a glucose unit. But now we don't build the tree and the leaf and the bark and the apple out of these glucose units. We have to connect them together to get them into a form that we can use structurally. And what that form is what is known as a polysaccharide. Now, it's not a monosaccharide, it's not a single sugar unit, it's not a disaccharide like sucrose, maltose, or lactose. Those are 2 sugar units. Polysaccharides are essentially nature's polymers. So nature has learned how to polymerize as well as humans. We talked about polyethylene, polypropylene, polystyrene, and so many others, maybe at the beginning of last week. Now we're looking at nature's form of polymers, and by far and away the most important form of nature's polymer is the so-called polysaccharide. And polysaccharides are polymers of the monosaccharide. They have 3 biologically important forms that we'll look at, and they differ by the way in which the glucose is connected together so the plants build the glucose, and then they connect glucoses together to make these 3 forms: cellulose, starch, or glycogen. So let's take a look at what is the fundamental difference between these 3 polysaccharides or combined forms. Now, really, the differences between them are quite subtle, but the differences between them are important enough that they have drastic differences in the way humans can process them. So first, let's start with probably for plants, the most important of them, and that would be cellulose. Okay, so this is the major structural material of plants. This is what leaves are made out of, cotton is made out of, this is what limbs and bark is made out of, this is what all of that is put together to build. Okay, so there we go. Cotton is about pure cellulose. And what does it look like? It looks like this. And this might not be very recognizable, but all that it is, in actuality, is a homopolymer of glucose. So the glucose rings, remember, are these individual 5-member, actually, I'm showing them here in 6-member rings. They are 6-member rings, sorry. 5 carbons in the ring and an oxygen. That's the way the glucose is. Then you got a carbon hanging off the side, just like you see there. And then when you connect these glucose units together, you get the polysaccharide known as sucrose. Now, it turns out humans have no ability to digest cellulose. So when we chew on cellulose, we don't have the ability to break it down. So what happens when we do, and does it mean we never ingest cellulose? Of course it doesn't. We ingest lots of cellulose. So what happens with the cellulose that we ingest? It passes through. So what we would refer to with regard to cellulose is we would refer to it as roughage. And so this is non-digestible by us. So it just kind of ends up passing through our digestive tract. And when it passes through, it comes out the other end. Now, is that a bad thing? No, that is not a bad thing, because as it passes through, it sort of sweeps out the tract, taking other things that might be left behind with it. So it's always a good idea to ingest enough roughage or cellulose in order to keep the system sort of swept out, if you like. Okay, now, as it says there at the bottom, there are other animals that have the ability to digest cellulose. Now, it turns out that they don't actually have that. It's the critters that live within the critter. So cows and goats and even termites have bacteria that live in their guts. And it's actually the bacteria living in their guts that has the ability to break the linkages between the polymeric rings and then use them as individual glucose fuels. Remember, in animals, glucose plus oxygen gives carbon dioxide, water, and energy to move. So animals break apart burn the glucose to make the energy to keep them alive. And in particular, goats, cows, and even termites have the ability to take the cellulose, process it down into individual glucoses, then use the individual glucoses to run their engines. Again, humans, while we can do it with other carbohydrate forms, including the one we're going to show next, we don't have the ability to do it with cellulose. Now, this one, however, we do. Now, I drew this cartoon. I kind of stole this and this differently, and it's really hard to see the difference between them. And I doubt if I would ask you to identify the differences between them. Very, very, very similar structures between starch and cellulose, but starch is a polymetric form that is used as the storage in plants. And so when plants, again, do too much photosynthesis, now, first of all, we should point out, not all plants create starch. Only certain plants make starch. What are some of the famous starch makers in the plant world? Well, potatoes are a lot of starch. They're not the only thing that's a lot of starch, and really nothing right now is coming to the tip of my head. What else has a lot of starch in it? But certainly potatoes have a lot of starch in them. It's, if you like, a storable form of cellulose that plants can use. They can later convert it into cellulose through some enzymatic processes. And starch consists of two kinds of polysaccharides that have some kind of linkages between them. So this is not, if you like, a homopolymer. It's a copolymer that makes starch instead of a homopolymer that makes cellulose. So amylose is about 70 or 20% of starch, and that's what we have the ability to break down in our mouths. We have an enzyme in our mouth known as amylase, and amylase, when we put starch in our mouth, starts to break the linkages between the individual or the combination polymer units, and we can use the individual linkages to fuel our system. So amylose is a straight chain linkage of the glucose rings, and that's okay by us. We know how to break those apart. Now, another thing that then happens with the starch molecules is that they begin to, as we say, cross-link. And the hydrogens of one chain of this starch will be attracted to hydrogens on the other chain, another chain of these starches, and we form what is known as a hydrogen bond. Now, this hydrogen bond is not really a share of electrons between them as much as it is a charge difference between them, a plus and a minus, almost like an ionic connection between various strands of the starch. So what happens is that this begins to coil up the starch chain, as we're showing here, and it becomes a helix. So the attractions of hydrogens for one another on various points of the chain turn this not so much into a straight chain, but into a coiled chain. Now, this coiling of important biological molecules is not only present in starch, but this coiling effect is also incredibly robust and incredibly important, even more important, we should say, in the messenger carrying or the data storage molecules that are used in the biological world. And I'm sure the initials for the storage, the data storage molecules are familiar to you. They are DNA, deoxyribonucleic acid. And of course, as I'm sure most of us are aware, the DNA molecule contains the code that tells certain units within us how to build the components that make us what we are. So there is a blueprint, if you like, to how to build a muscle tissue. There's a blueprint of how to make your kidney work. And this is stored in the connections of various so-called nucleic acids in a particular pattern that tells these replicating cell engines how to put certain components together, certain proteins, certain amino acids, and very complex collections of molecules in certain shapes and sizes in order to make functioning materials such as organs and such for living systems. These coded messages, just like we see here in the starch, because of, again, hydrogen bonding between various parts of the strand of the molecule, the lengthy molecule begins to coil up upon itself, just as we see here in the way these carbohydrate monomeric units are coiling up on themselves to make the structure for this starch material. Okay, so amylose amylopectin, is the other form of the way starch is constructed. And it, as you can see below there, has some kind of branching to the chain. Instead of being a straight chain, there are certain branches off the chain that are connected together. And this amylopectin is a branch chain, as I said, instead of a straight chain. And this also is the way nature has figured out to polymerically connect individual glucose units to build a polymeric form that can be stored in, again, plants and various regions of plants, and then used later if it is required to build some cellulose. All right, so unlike cellulose, humans have the proper enzymes to digest starch, and starch is first broken down into what we call complex carbohydrates. That's probably something you've heard of at one time or another, a complex carbohydrate known as a dextrin. And then eventually through other enzymatic interactions, we break it down to maltose. And then finally, we know how, through the wizardry of our animal structure, to break it down into glucose. Then the glucose can be used in respiration. Remember, glucose plus oxygen makes energy, most important, plus carbon dioxide and water. Okay, so this amylase, as I mentioned, that is an enzyme in your mouth. And when you start chewing on certain starches in your mouth, the amylase begins immediately to break the starches down. So when it's on its way to your stomach, it's already softened to a great extent and began to extract to the complex carbohydrates so that the digestion process can yield fuel from the starch that you ingest. Okay, so what else do we have to say here? So we get about 50% of our nutritional calories from this mechanism. Surprisingly, we get a lot of our fuel from this complex carbohydrate process that we're talking about here. Okay, now then the other form of connected polysaccharides that are built are what are known as glycogen, and this is what animals make. When you ingest too much of these carbohydrates yourself, one thing that we do with it is make it into a form that we call in animals, on the animal side of the tree, glycogen. Now again, glycogen is a polysaccharide. Glycogen is made from monosaccharide units of glucose. And once again, instead of being in a straight chain form, it is in some branch chain form. Now again, what that means is it has great similarities to what we saw on the previous slide, but it is not put together exactly in that same form. That's how trees or plants do it. This connection of the monomeric units to form this polysaccharide is in particular the way that humans do it, or not just humans, animals in general, living animate systems do. So it's a polysaccharide stored, as it says here, in muscle tissue, stored in liver tissue, and it's used to maintain the proper glucose levels. So this is, if you have problems with your glycogen levels, then you're going to have problems with your glucose level in your blood. And so people with diabetic conditions have some upset regulatory processes that are, believe me, studied but not well understood. If they were well understood, then we would have conditions such as this under better control. We can certainly control them, but we can't eliminate them and remove them. That's for sure. So believe me, there's a lot of emphasis on understanding these kinds of processes, but they're, again, not completely well understood or regulated, or we don't have the proper medications or rectifying processes to eliminate these issues, even though humans have dealt with them for hundreds of years now. And again, the issue that it boils down to is that certain individuals can't properly regulate their glucose levels, and it leads to all sorts of issues collectively placed under the umbrella of what is known as diabetes. Okay, so the structure of glycogen, as you can say, as I said earlier, very, very similar to this amylopectin. It's got a branched monosaccharide structure that forms a polysaccharide, as we say here, and it's even more branched than is done in the plant world. Okay, so let's move on to some other molecules of importance, certainly again in terms of this storage importance, and these are what collectively are known as lipids, and lipids comes from the Greek word for lipos, and lipos is the Greek word for fat. So a lipid is a generalization of a fat molecule, and really it goes into two classes. A lipid is either a fat or an oil, literally depending on whether it's liquid or solid. That's the entire difference between them. So liquids are nonpolar and are, as it says there, lipids, they tend to be nonpolar, but it says they're not very soluble. Sorry, I wasn't really looking at it correctly. I thought it said they are, but fats are not very soluble in water. And I'll show you why they're not very soluble in water when we look at a picture or a cartoon representation of the structure of a variety of fat molecules. Now, what this means is what we typically call fat when we try to wash it off, we call it greasy. Greasy is a slippery feel to it. And when you dump or you rub water on it, the water doesn't seem to have any impact on the greasy feel that you might have if you get some of this type of material on you or on your clothing. So when we have issues like this, our universal solvent invariably is water. And water is this polar material made up of just H2O, of course. And when you inundate other polar materials with water molecules, the polarity of the water draws them along with the water, and the liquid flow of the water just sort of drags these impurities away with it. And water is universally used to wash away components that are contaminating something that you want to get rid of. Now, if you try this same approach with lipids, the problem with lipids is because they're nonpolar, they don't interact or stick or bind with water molecules, so therefore cannot be drug away by water molecules. So we have to use a trick that maybe we will see in a later slide here to help us change the fat into a form by which we can now wash it away with water. So maybe after a little introduction to the idea of lipids here, we can look at how we have figured out to wash away greasy items. Okay, so lipids fall into three types of categories, just kind of like starches did. There's going to be fatty acids, there's going to be triacylglycerides, glycerols. Glycerides are a little different actually than glycerols. And then there are steroids. So you may or may not have heard of all these terms. We're just going to briefly show the differences between them. We're not going to get gory involved in this. Again, if you really want to get into the guts of these topics that we're discussing here, we have other classes in the chemistry building that you can take that delve into these things in much more depth. Here, we're just trying to get some appreciation for these types of molecules and at least certainly from a consumer standpoint or a dietary standpoint, why these things have the kind of effects they do on us. All right, so let's just say a few things about these three different forms of the lipid tree, if you like. Let's start with acids, and in particular, what we will call a saturated fat that results from these. So a saturated fatty acid is an alkane. That's an important designation. We talked about this concept when we were talking about organic chemistry some time ago. An alkane is a carbon-hydrogen organic molecule where the carbons are only singly bonded. And remember what we mean by an unsaturated fat or an unsaturated hydrocarbon. That's when you have multiple bonds. So in an unsaturated hydrocarbon, there can be alkenes or alkynes. Saturated hydrocarbons are alkanes. Now, what makes this a saturated compared to an unsaturated? When you maximize the hydrogens, there can be no multiple bonding between the carbons. So that's the big trick. Only single bonds between the carbons, and then the rest of the connections that the carbons have are to hydrogens. So unsaturated fats have multiple bonds and have fewer hydrogens. That's the big designation. Now it turns out that unsaturated fat versus saturated fat have different ramifications in our health issues. And I will honestly tell you that it changes very regularly, whether saturated fat is good for you, unsaturated fat is good for you, polyunsaturated fat is good for you. There's continuing debate among the nutritionists as to whether or not which of those forms of the fats are of particular are more beneficial than the other. Again, since I am not an expert in these differences, I'm not going to weigh in on whether the saturated fat is better for you than the unsaturated fat. You can read your own literature on that and make your own decision, because again, I will mention that the dietitians and the food industry has gone back and forth over which is better for you and which can lead to a healthier lifestyle. Okay, so again, a saturated fat is alkane bonds of the carbon. So here are some various saturated fats and their names and where they come from. So if you have lauric acid, you will note it has this general form. Now, let me point out to you what we're saying here. This is an abbreviated structure. Remember, an alkane in general is CH3, CH2, 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 CH3. The ending was always a CH3. Well, this is a little bit different. It begins with a CH3, as it should if it's an alkane. Then there is a bunch of CH2s linked together. So you'll note in the parentheses of the middle there, I'm showing CH2 and outside that 10. So of course what this is implying is that you're going to link 10 CH2 units to that end CH3 unit. Then on the right is something a little different that we might not have seen before. COOH, which is an indication that there's a carbon connected to two oxygens ending with a hydrogen. This is a very common connection of atoms in the organic world. It is what makes a so-called organic acid. So all fats contain this connection. All fats contain the so-called organic acid group. So technically, all fats should more correctly be called fatty acids. So all of these are acids. The left part of the molecule is the alkane, and then the alkane is always connected to COOH, and this connection to COOH is what makes it into a fatty acid. Now, the O's that are connected at the right side are very polar. So the right side of this fatty acid is always going to exhibit attraction to other polar molecules. However, the left side of this molecule, where you see all the C's and H's connected in a straight chain, this region of the fatty acid is very non-polar. The C and H's are distributed in space so that charge is evenly spread around. So if I had drawn one of my structural pictures for this, like I did for the sugars before, what you would see is the left side of this would be all green. Because remember, green meant even distribution of charge. The right side of this, where the COOH is, would be all red, because that is the region of the molecule where all the electrons are piled up. So again, the long straight chain alkane part of the fatty acid is nonpolar. The COOH region of the fatty acid is polar. Okay, so where do we find lauric acid? You find it in coconut. So this is the way that in the plant world that coconut makes coconut oil. So coconut oil is essentially this material that we're calling lauric acid. Now the next one is muriatic acid, as you can see here, and it has this formula. Notice the only difference between lauric acid and muriatic acid is that the long chain alkane part of the molecule has 2 more CH2 linkages. So the alkane part of this has 13 carbons then hooked to the organic acid, as opposed to the lauric acid, which had 11 carbons hooked to the organic acid connection. Now, where do we find this material? You would find this in nutmeg. I'm sure it's also present in other parts of the plant world, but this is one place in particular. You would find this form. The next one is palmitic acid. Again, all we're doing is making the chain even longer. You would find this in palm oil. Then we have stearic acid. Stearic acid, you can see, again, even a longer chain. We find this in humans, or actually not just humans. Stearic acid is present in cows when we ingest fat, gristle, or whatever you want us to call it, from a piece of steak. This would be the form of that material that you're ingesting. So all of these fatty acids contain the so-called carboxylic acid functional group. All of them have the COOH, which is the actual component that makes them behave acidically, and this is their source of polarity. Now, I also want to mention there are many other forms of fat, than these 4 that I'm listing here. I'm only listing these ones here because these are the so-called saturated fats. All of these have only single bonds between the carbons. As we move forward here a little bit, we'll take a look at some other forms of the fats that are possible as well. Okay, so this says that this is the alkane part of the molecule, as I indicated before. This makes them very insoluble in water. This region of the molecule is the stuff that the greasy part of these things would keep from allowing them to be dissolvable in water. Okay, now let's look at some unsaturated fatty acids. So now what is the difference between them? Well, it's whether they're mono or polyunsaturated is one difference in them. If they're monounsaturated, they have only one multiple bond between the carbons. If they're polyunsaturated, they have at least 2 and more likely more double bonds between carbons. So these unsaturated fat forms are also very common. Let's just look at some names of them and where they might come from. We have this one at first, and it has this formula. Now, I want you to look, I'm gonna get my glasses out so I don't mess up what I'm talking about. The I can't even say the name very well myself. Palmitoleic acid. You will note that at first there is alkane connections. So I have CH3, CH2, CH2, CH2. There's 5 of those in a row. Then there's a double bond in the middle. Then we have, again, 8 more carbons in an alkane connection. So it starts off on the left with 6 carbons in an alkane form. Then there's a double bond kind of in the middle, not quite, but kind of in the middle. Then we conclude with 8 carbons in an alkane form, and then you have the so-called carboxylic acid connection on the end. Now notice there's a single double bond in this particular molecule. So this is one of our mono unsaturated fats. And this is what is the source of fat in butter. So it comes from dairy originally. So if we have oleic acid, this will be the form of oleic acid. Again, you can see this is a mono unsaturated fat. Again, it begins with a, looks like an 8-chain alkane, then a double bond, and then it has another 8-chain alkane followed by the organic acid group. This one is present in either olives or corn. So we process this to make corn oil. We process this to make olive oil, of course. So if you purchase those things, this is the material you will be getting there. Okay, now we have linoleic acid. And again, it looks like it is also a monounsaturated fat. That would be what you find in soybeans and sunflower. So this is often sold also in the grocery store. Soybean oil, sunflower oil. You can buy this stuff as well. And then finally, we have, the difference in the name here is very subtle, as you can see. Wait, that looks like the same name. I guess I messed up with this one. I'm sorry. But you can see for this one, it's even a longer chain connection between them. And this one is present in corn. All right, so let's take a look at some triacylglycerols and what they are. So this is the major source of animal storage. This is how we hide this stuff in our bodies. So these are made from glycerol hooked together with fatty acids. So this is what we often call fat, not the fatty acid. So we will take those 2 units of those things we saw on the previous slide, hook them together to make them into this form. So here's what we say about them in regard to where we find them. We find them in, as we say, meat, milk, butter, cheese, stuff like that. And the only difference between a fat and an oil literally is whether or not they're liquid or solid. So I think I actually have some of them on this slide here. Let me just show you. We have the various oils that we often use, which are mixtures and way these things are hooked together and stored. And so notice that some of them have temperatures where they are above 0 and some of them are below 0 where they melt. Now, of course, again, let's think of what we mean when we say they melt. When they melt, that means they're changing from a solid to a liquid. So if you look at the first one, it says beef tallow, which is beef fat. Beef fat melts at 42 degrees C. So it's before it melts, it's a white substance. I'm sure we've all seen it. And if you heat it up to 42, it will melt. Now we're on that temperature scale. Let's look at some common points on that temperature scale. The human body is at 37 degrees C. That's the temperature that we exist at. Of course, water boils at 100 degrees C. Room temperature is like 25 degrees C. So beef tallow would be solid in room temperature. It'd be solid at body temperature, but beef tallow would be liquidy if you put it in boiling water. Now butter, it says there, has a melting point of 32 degrees C. Once again, the human body temperature is 37 degrees C, so it would be liquefied at human body temperature. Room temperature is like 25 degrees C, so you're getting close to the being liquid at room temperature, but it should be solid at room temperature. Coconut oil is right at room temperature, and that it is an oil typically at room temperature, but if you cool it even slightly, it should solidify. Olive oil you see. Now, let's remember that water freezes at 0 degrees. So if you put olive oil on ice and maybe cool it a little bit colder than the freezing point of ice, you can see you could solidify olive oil. Now the remaining oils are actually pretty far away. They'd be hard for us putting them in our freezer. I don't think we could solidify canola, soybean, sunflower, safflower, or corn oil. We'd have to have a little bit colder situations than our room in order to solidify them. But again, what I would like to point out here is the only difference really between a fat and an oil is whether or not it's liquid at room temperature. So, of course, beef fat and butter and even coconut are really right on the edge of being fats. And then everything below that, olive oil, canola oil, soybean oil, sunflower oil, these are all oils because they are liquid, literally at room temperature. Now, otherwise, structurally, they're very close to one another. They have slightly different length of their alkane chain, but not very much else different between them. Okay, so let's just look at some forms of the so-called triacylglycerols. This is when you hook 3 glyceride units together. So if you do things like this connection, you make what is known as a triacylglycerol. So you cook 3 of the stearic acid, fatty acid molecules with 3 connections of a glycerol molecule, and then they kind of spit out this water between them, and it makes this connection. And this is what we call a triacylglycerol. Now, it turns out that these things are also very important in living systems. So people don't have the proper regulation of their triacylglycerol levels in their blood. You get too much of this stuff in your blood gets a little too viscous, and then you have trouble, your heart has trouble pumping this stuff around because you have too high of levels of this kind of stuff in your blood, and your heart has to work too hard. You have very elevated blood pressure if you have too high of levels of things of this sort in your blood. So there are many medical things that we currently have to combat the level of triacylglycerol that you might experience in your blood system, so that we can keep our blood system working efficiently and not stress our hearts out too dramatically. Okay, so again, the idea of saturated fat and unsaturated fat boils down to the fact that you either have multiple bonds to the carbons with the hydrogen, or you have all single bonds between the carbons in the molecule. If it's an alkane, it is a saturated fat. If it is an alkene or alkyne, then it is an unsaturated fat. And if it is unsaturated, it can be polyunsaturated if you have more than one connection between them, or if you have only a single one, it could be what we call a monounsaturated fat. Okay, so again, they go back and forth on this, whether the polyunsaturates are good, the saturated are good, the monounsaturated are good for you. I just can't keep up with it, whether or not, or which of them is good for you, which of them isn't good for you. So you can look into that yourself if you like, is what I would say. Okay, so here are some ratings of the various fats that are out there, and how much of them are present in various things that we ingest. I thought I put some, I put some things up here. So this shows you if you have these various types of oils that are commonly used by us, what percentage of them are saturated fat, what percentage of them are unsaturated fat, and what percentage of them are monounsaturated or polyunsaturated. So you can see like coconut oil is essentially all of the saturated fat type. You can see that cod liver oil and olive oil in particular is mostly monounsaturated fat. So a lot of people these days, I guess, would say that the canola and the olive oil are good because they are high in monounsaturated, kind of moderate in the polyunsaturated, and things like coconut oil, very high in the unsaturated fat, maybe not so good for you. I think that is pretty much the way these things are going at this particular juncture. All right, now just I don't think I want to skip this part because I want to finish these ones up. I'm just going to skip all the things about hydrogenating. I'm going to skip the things about, no, maybe we will say this because I did want to mention how we ultimately take greasy things and take care of them when we try to wash them off. What we do is we make what we call a detergent molecule. And a detergent molecule is made in a process of saponification. So when we take a fat and you heat it with something like sodium hydroxide, which is called lye, we get this molecule that turns into a form that we can now use as what we know as a detergent. So let's talk a little bit just about how a detergent works. Now, what the point of a detergent is, it's going to take something that shouldn't dissolve in water and make it dissolvable in water. Now, the problem with fatty things, you get some French fry grease on your hands, you try to wash your hands, and you can tell that you're not washing the French fry grease off very effectively. So maybe you rub some soap on your hands, and now you find it a little bit easier to get the grease off your hand because you're using the soap. So what is the soap doing? Well, the soap kind of bridges the gap between the parts of the molecule that are nonpolar and making it into a part of the molecule that is polar. So again, here's the problem. Water is polar. And water as a solvent to wash stuff away only can wash away polar entities. Fat and grease is essentially nonpolar. Now there's a little end of it that's polar, but the vast majority of the chain of the fat is nonpolar. So what the soap will attempt to do, or a detergent in general will try to do, is to make the molecule so that it will dissolve in the water. So let's just look at a cartoon of doing this. I'm not so much, I want to just go right to the cartoon of showing you how to do this. Here's how a detergent will pull this off. What it does is that there's a part of the molecule which is highly nonpolar. It will interact with the soap molecule so that the nonpolar end of the soap sticks to the nonpolar end of the fatty molecule. Then the other end of the soap molecule is polar. So it points a polar end away from the fat molecule and a nonpolar end in towards the fat molecule. So the polar end sticking outward can now interact with the water. The nonpolar end sticking inward can interact with the fat. So by encasing and encapsulating the fat molecule in this way, now if I dump a bunch of water on it, the polar ends sticking out are drug along with the water and can be dissolved in the water and washed away. Okay, so we're going to quit there, and I'm not going to do anything else on these slides. I might ask you a little bit about the soap thing, maybe. I don't even know if I'm going to do that, but we will have in our next unit some questions on some of these biochemical things, in particular the carbohydrates that we talked about, and you'll be able to look them up on our slides. Remember, please go to the Moodle and complete test 3 part 2. It closes tonight at 11:30. So make sure you do that by 11:30 tonight. So until next time, peace out.